Yeah. Scott's doing some pretty amazing work and has been doing some pretty amazing work at UBC for a number of years. And I saw a presentation that that his team made about one of the latest studies that he's done. And I thought it would be really cool for him to just give us a little update on what he's been working on. So without further ado, Scott, you can take it away.
Yeah, can you make me co-host so I can share my screen?
Sure.
Maybe I can. Okay, I'm good.
How many co-hosts am I allowed?
I think I'm good here. Can you see this?
Yes, we can.
Then you are good.
Yeah.
Okay, well, thank you. I appreciate the opportunity to speak again to your group. This will be about a 15-minute presentation. There's some data heavy slides, but lots of pretty pictures as well. So the study that Kevin's referring to was recently published, and you're all welcome to get a copy of it. The link is down at the bottom. It also got picked up by in the news recently, so you may have seen some talk about it there. The title of the study is Exceptionally High Mortality of Adult Female Salmon, a large-scale pattern and a conservation concern. And I just want to thank a lot of people before I move too far into here that have helped with this. I have several co-authors, Nolan, Erica, Tony, Steve, and David, who have been with me for several years, and then many grad students and technicians who have helped collect data for what is basically a 20 to 30 year study. So I want to thank all of them up front. I'd also start with this little cartoon. This was appeared in the Vancouver Sun about 10 or 15 years ago, but it's so appropriate now. And I don't know if they realized when they drafted this cartoon that the salmon that they're talking about not being able to keep going was the female, and the one that was able to keep going was the male. And this is actually something that is the core component, the core thesis of this research is indeed that females are struggling a lot more than males these days. So a little bit of the design of the research. So over the past few decades, we've been conducting lots of different studies looking at migration mortality of adult salmon. And what I've done is I pulled together the key results from 19 published studies. And these 19 studies gave me 40 situations, 40 experiments, 40 trials where male and female mortality could be directly compared. Those 19 studies all involve sockeye, but there's also 2 or 3 other studies that I report on that involve coho and chinook. So this isn't just a sockeye issue, although that's where a lot of my research has focused. Give you a bit of a spatial perspective on where this science occurred or is occurring. This rather complicated looking map has lots of different colors and numbers on it. The numbers just reflect a code representing a particular published paper. So don't worry about the number, but what's most important are the colors. And so the blue dots reflect studies that involve telemetry tracking. So where we put transmitters into fish. And so this would have been tracking a particular population at some point along their migration route that was heading to a natal area, and the natal areas are indicated in blue dots. We had many laboratory studies as well, some conducted on the very same populations, and these fish would have been intercepted along their migration to where the red dots were, and they would have been taken to the lab and they would have been examined there. And then the purple colors reflect codes for populations where we were able to obtain spawning ground sex ratio assessments. So this is something that is not done with every species, but sockeye has a really rich data set in terms of looking at sex composition on spawning grounds. So we were able to take a look at historical data obtained by DFO to see what's going on over time with sex ratios. So I think I'm going to start with just the take-home message. So what did we find? Well, the first thing we found was that female mortality across all the studies averaged about 2.1 times greater than males. In some studies, it was up to 8 times higher. There wasn't any instances where male mortality was greater than female in any of the studies. Female mortality was highest when the migration conditions that those fish experienced were the most challenging. So for example, if they were encountering either high or turbulent flows, high temperatures in the field or in the lab, confinement, or even handling, both in the lab and in the field. All those conditions set the stage for higher female mortality. And also that the scale of the mortality was greatest, that the differences were greatest for females towards the end of their river migration. So when the fish were getting more and more mature. And as I mentioned a moment ago, we were able to look at sex ratios. And so what we've been seeing now when we look at this for several populations is relatively fewer females now on several of the key spawning grounds, at least for Fraser sockeye, where we have data. So I just want to show you what some of that looks like. So these are three graphs that show the percent of females in a population. So the percent of females relative to males. And these are three early run populations. They're all heading to the early Stewart area, which is about 1,000-kilometer migration from the ocean. And what you can see is that this declining trend, the red lines over time. We used to have historically back in, before the 2000s, way higher numbers of females on spawning grounds. In some cases, you can see 70% or higher of the fish were females, but that has been in decline. And in recent decades, either the ratio is now getting close to 50-50, or in some years, as I'm showing here, we're now seeing many fewer females than relative to males on spawning grounds. And this was for early populations. This is a very similar looking figure, but it's three different populations. These are late runs now. These were ones that migrate late in the summer, early fall. These are going to the Weaver Creek area, not too far from us, Adams River or the Lake Shuswap. And again, you see the same general declining pattern that we saw with the early runs as well. Now, there were some populations that didn't show these trends, and those were interestingly, the ones that migrate in the summer runs. And so the summer runs might be a bit more resilient to some of these consequences, but as we found in a lot of our lab work, that wasn't necessarily the case, but we're not necessarily seeing this appearing on their sex ratios, but we certainly see it with the early runs and the late runs. To give you a bit of an idea of how we do some of the research, and I think when I spoke with your group a year or more ago, we talked about telemetry tracking, and certainly we do this with the adults, just like I was talking about it with your group earlier with juveniles. And we collect fish either in the ocean, the estuary, or lower river reaches. We would use either purse seines, as you see here, for ocean capture, or beach seining in river sections. And then those fish would either be used in telemetry tracking, or we'd take them to the lab. The one thing that makes all this research really unique is that we always collect blood samples from these fish. And so you can see us here collecting a blood sample. And this gives us a lot of information on their initial physiological state. We're interested in the ion concentration in their plasma. We're interested in stress hormones, but this allowed us also to look at reproductive hormones. And so with knowing the reproductive hormone levels, we can provide a sex determination for these fish, even though physically you wouldn't be able to tell sex by looking at them. And few investigators really ever look at sex as part of their ocean or river tagging, and it's really difficult to identify sex without blood work. So this is probably why this phenomenon has not been discovered before. But certainly when we start looking with a careful eye to the sex aspect, we start seeing this occurring repeatedly, this phenomenon. When we're doing work in the field, we put the transmitters usually into the stomach of the fish because they're not feeding any longer. So this is just showing us popping a transmitter with a little plunger down the throat into the stomach. It's quite harmless to them. It takes only a few seconds, and then we can release the fish with a secured transmitter inside of it. So I'm just going to show you some results from three studies, or three types of studies, to give you a feeling of the sorts of results and the study approaches we've taken. So what you're looking at here on the far right, you can see is a close-up of the Thompson River watershed. So this is the Fraser River in brown, and then the Thompson River in blue. And this was a telemetry study that we did several years ago where we're tracking adults to their spawning grounds. That's what the red dot is. And we were able to identify at least 100 of these fish as females and almost 100 as males. Some we couldn't identify because we either didn't have the blood samples or the results were inconclusive. But for ones where we had the sex ID, we were able to look at this relationship. And so I just want to tell you that we separated the study area into zones, and we were able then to calculate survival in between these dots. So we have sort of a lower section of the migration, a middle section, and an upper section. And And so now I'm just going to show you what we found for those 3 different sections in terms of how well these fish survived en route to their spawning grounds. And I broke it up by sex. And so it's a really strange survival index. Don't worry about I've calculated it per 83 kilometers of migration, but it's all relativized. But what's important to note is the dots are males and females. Males are blue, females are red. And the letters just reflect whether or not the dots next to each other differ. And if the same letter means they don't differ. And so as we moved across the migration range from lower, middle, and upper, we don't really see any changes in survival rates. Now, these fish were migrating in this particular portion of the study at about 14 degrees. No difference in survival between the males and females at any of these sections. The overall survival rate for females was about 86% to get to the spawning grounds. The overall survival rate for males was almost 100% to get to spawning grounds. However, it's a very different pattern when we looked at the survival rates when temperatures were warmer. So when the temperatures were 19 degrees in the river, we don't see any difference between male and female survivorship in the lower river. We start seeing differences emerge in the middle portion, and we really see those differences emerge when they get towards the upper portion of their migration. And the overall survival rates from start to finish, only 50% of the females survived under 19 degrees. And over almost 80% of the males survive. So there's 2 take homes with this. We see much higher female mortality closer to spawning grounds. And just overall, we see much higher female mortality across the entire migration. So this is one of the first studies that really got us thinking that there was something going on with male versus females. And then we followed up with many other studies along these lines. I'm going to show you now some telemetry tracking studies we've done looking at fish moving across a dam on the Seton River. So on the right, you'll see a map. Here's the blue dot showing where the Seton River is. It's right near the town of Lillooet. And so this dam is just a few kilometers upstream from the town. There's a fishway located right on the dam here. You can see where I'm pointing to the entrance to it. So the fish have to find this fishway. And here it is in another picture. And then they have to migrate up the fishway over the dam, and they travel about another 40 kilometers to get to their spawning grounds. So we studied this system over about a 10-year period, and I'm just going to show you some of the results of what we found between the sexes in terms of their survival rates. So in all cases, fish were captured around the fishway or just below the fishway. We tagged them, we released them, and we let them migrate back up, find the fishway entrance, and migrate through and on their way to spawning grounds. What we found is that in all cases, female mortality was almost always twice or higher than twice that of the males. So in 2007, mortality to get to spawning grounds after getting through the fishway was about 60% for females, is only 29% for males. 2012, we did the study again, almost an identical result. We did the study again in 2013, almost an identical result. So we start seeing these consistent patterns over and over again of females having much higher mortality than males trying to get through a similar area. Other types of experiments we did where we were able to look at this sex effect was in the lab. And so we've done lots of lab studies where we've tried to look at the effects of getting captured in fishing gear and getting out of fishing gear. And this is an experiment we did on fish that were heading to the early Stewart system. Up here, this big red dot on the right. They were captured in the lower river and taken to our lab at Cultus Lake. And they would have been en route to a 4-week migration. And we basically grabbed them and held them for 4 weeks in the lab. And during that period, we entangled them and released them from a gill net. And then we monitored them and saw how well they survived. And so you can see us here letting fish get entangled in a gill net. And then you can see the gill net marks on the fish here. And then we put them back in the tanks and we saw what happened. And here's the large tank compound that we have where we do some of this work. And what we found in this case was that even if you didn't touch the fish, even if you just put them in the tanks and just monitor them over the 4-week period, the females still survived much more poorly. They had like 23% mortality compared to males. This is a confinement effect, a captivity effect, a handling effect. And so females are much more sensitive to that than males. But when you layer on additional stressors, you really see the differences magnify. So when you put the fish into a net for 20 seconds and remove them, females only survived, had 70% mortality compared to males that had 44%. if you gave them a 20-minute entanglement and then released them, almost all the females died before the 4-week period was up, 90% mortality, whereas 75%, a much lower value of males perished. This is just one example of a type of experiment that we've done several times. We did it with Chinook, we've done it with coho. We found, again, similar sorts of differences between males and females in these lab studies. So brings it all together. Here's a summary of the 19 studies involving 40 different experiments. So what you're looking at on this axis here is a ratio of the percent female mortality to percent male mortality in 40 different experiments. Each dot is that ratio. The black dots are studies we did in the lab. The white dots are studies we did in the field. And the just treatment number is just numbers of it's just a category of a particular treatment. It's just an accounting of the 40 different experiments that we did. So what you can see right away is that in every situation, there's not one situation where male mortality exceeded female mortality. There was some where they didn't differ; they were similar, and in those conditions, those were relatively benign environmental situations or benign experiments. But once we exposed fish to various stressors, like the ones I showed you earlier, we start seeing these differences between the sexes emerge in survivorship. And you can see, typically it's around 2 or even higher in terms of twice the level of mortality. In some cases, remarkably higher mortality in females. This was a laboratory study, and they certainly didn't do so well compared to males. I think they were just held under high flows in troughs, and the females did not do as well as the males. But it's a very consistent, repeatable pattern across the board. So then it begs the question, why? Why is this happening? And that is a real difficult nut to crack. And we've narrowed it to 4 leading hypotheses and probably 1 or 2 probable causes. One of the likely explanations or leading explanations is they may be running out of gas. They could be exhausting their energy stores. They all have a limited amount of energy, as you know, when they start their migration. They're not feeding. They're going to all die eventually when they get to spawning grounds, but some might be utilizing those reserves quicker than others. And we know that females deplete their reserves quicker. And the reason we know this is because the size of their gonads, as you're well aware, is so much bigger in females compared to males. In sockeye, it's about 15% of their body mass is gonads. In males, it's only about 2%. So that means they have to put a lot more energy from their reserves into gonads. So that means a lot less available for them to be swimming. On spawning grounds, we note that females deplete way more of their body energy as well in their basic activities. So about 27% of their energy gets depleted relative to only 18% on males. So there's lots of evidence that females, particularly those that have to migrate 1,000 kilometers to get to spawning grounds, perhaps they're running out of energy reserves at a greater rate than males. So that's one possibility. Another possibility is that they're just much more stressed, and this physiological stress is pushing them over the edge. We do know that females have much higher endogenous stress levels. We measure this with plasma cortisol. Cortisol is an indication of stress. And fish mobilize cortisol. It's actually to their advantage to have cortisol mobilized when they are stressed, because it does allow them to access energy reserves better. But when levels of cortisol get too high, it can actually have negative consequences. One of those negative consequences, it actually might impair their ability to get access energy, and it would impair their ability to clear waste. And just to give you a sense of just how much higher the cortisol is in females, it actually rises to 4 times their baseline level during the migration, whereas in males, it only rises to 2 times their baseline. So females are much more stressed from a plasma cortisol level than the males are. And give you a perspective of just how high that level is. If you were to track fish through Hell's Gate, here the narrowest and perhaps most turbulent water that fish could encounter, those levels of cortisol that fish experience migrating through here for, let's say, a day is what the females are expressing throughout their entire migration. So it puts it in perspective just how stressed females can be. Another possible explanation is that they're sick, or they're sicker than the males. Certainly, we've looked a lot at infections, and there is a link between cortisol levels and an infection. And these high female cortisol levels could make them more immunosuppressed. In other words, they might be more susceptible to picking up bugs. We also know that as females senesce, they're unable to fight off infections. And then, to make matters even worse, we've done some pathogen work recently, and we found that females may even start their migrations with higher pathogen loads, just a naturally higher pathogen loads, different bacteria and microparasites. So it's possible that, that the combination of being more stressed and the fact that they are more immunosuppressed might be contributing to their higher mortality. And lastly, but perhaps most importantly, and the theory that we think is probably the most likely one, or at least the likely driving one, is that they're dying from broken hearts. And I use that term kind of jokingly, but not completely. We think they might be experiencing cardiac collapse. So basically, they're having heart attacks. Females do have poor cardiac performance right from the get-go relative to males. They have a much smaller, what's called metabolic scope. That means the ability to do work using their basic metabolic processes. And they don't have the same ability for venous movement of blood out of their heart and through the rest of the body. And to top it off, they have smaller hearts than males, about 13% smaller, given their size. And so their cardiac capacity is indeed lower than that for males. Now, when you tie that in with the fact that their gonads are getting really big and they have to keep those gonads oxygenated, that means pumping blood to their gonads, then there's an extra drain on their metabolic abilities because now energy has to be diverted and oxygen has to be diverted to the eggs. So that's that much less that they have available to them to swim and migrate. So they're starting with at a lower level in terms of their metabolic and cardiac abilities. And now the gonad development issue makes it even harder. And we suspect this is why we see this effect of females having higher mortality closer to the spawning grounds, as in terms of the highest effect has a lot to do possibly with the fact that gonads are getting most developed by that point, and they have to really pump out a lot more oxygen to those gonads at the same time as they're struggling with higher temperatures and higher flows and having the reduced capacity to cope. It's probably not any one of these four hypotheses. It's probably combinations of them, but certainly this one is a leading candidate, we believe. And just to wrap things up, I've been asked a lot, well, what can we do about this? Is there a way that we can improve the situation for females? So here's one example of how we have actually been able to improve the situation for females. And it was not necessarily intended to benefit females more than males. It was intended to help all fish, but it turns out that females benefited quite a bit from this. And this was to modify how this particular dam operates. So this is the Seton Dam, the one I talked about a few slides ago. So just to remind you, the fish have to migrate up here. They have to get through this turbulent water, get into the fishway. They migrate up the fishway into Seton Lake and then through Anderson Lake and to their spawning grounds. This is what it always has been, the operational procedure at this dam. That's why it's called the routine operation during the sockeye migration. We noticed that fish were having difficulty getting into the fishway. You can see how turbulent the water was. They would get through, but to get through, they had to exert a lot of effort. And we used a really high-tech type of transmitter that gave us information on their tailbeat swimming speeds. So we knew how hard they had to work to get through this whitewater. And some of the modeling we did suggested that they were working so hard that they were building up large oxygen debts in their tissues. And it was the females, it turns out, that built up the largest oxygen debts. And it was the females that perished later on during the migration in a latent sort of effect. So these females weren't able to recover from working so hard to get through this turbulent water. So we thought, well, what if we just made it a little less turbulent? So we worked with BC Hydro and the local First Nation to try some experiments in a couple of years where we basically moved where the water came from just over a few meters. So it looked like this. And so now we have a much easier pathway for those fish to access the fishway entrance. Turns out that that alternative flow release improved the rates of fishway passage and subsequent survival to the spawning areas for both sexes, but it was the females that benefited the most. So there was a 25% increase in females getting to spawning grounds and a 10% increase in males. So this was a win-win-win, really. Everybody came out ahead on this. BC Hydro was happy because it didn't really affect them in a big way, because we weren't talking about changing how much water was coming through the dam, just where it came from. So this is now a regime that they now operate routinely during the sockeye run up in Lillooet. So again, just to revisit the take-home messages, female mortality averaged over twice as high as males, and up to 8-fold in some cases. The mortality was highest when migration conditions were the most challenging. So they had the highest or turbulent flows, the highest temperatures, they were confined or they were handled. It was observed to be highest towards the end of river migration when they're maturing. The likely causes seem to be energy exhaustion, cardiac performance, stress, or other immune factors. And I guess the unfortunate take home, though, all this is, female specific mortality is going to become even more pronounced in the future, because the changes that we're expecting with climate change are going to be making rivers warmer, and in some cases, altering discharge patterns, making flows even higher at certain times of the year. And unfortunately, those are the two factors that we know are going to lead to higher female mortality relative to males. So hoping that that doesn't come to fruition, but it's likely to be the case as we move forward in time. And with that, I'll end there. So thank you very much for giving me this chance to talk to you, and I hope I didn't take too much of your time of your meeting. I'm happy to answer any questions that you might have.
May I ask a question, Scott? It's Ruth here.
Absolutely, Ruth.
Thanks. Hi. I was surprised to learn that you were using blood sampling, like physiological characteristics, to determine sex, because I thought that morphology would give you that answer by the time they got to the spawning grounds. What's that about?
Well, they would, but we're doing our work, weeks and months before they get to spawning grounds.
Ah, thank you.
So we've, a lot of the work where we're tracking fish, we would capture, and I'm going to get rid of the screen so I can see your faces. 1 second. So I'm going to stop share here. There we go. I can see you now.
We can still see your screen.
I know. I'm going to try to shut that down here. Gotta. I think I know how to use Zoom. Nope, that's just up share. There we go. Most of the work we do or have done, we are accessing fish that are either still in the ocean, in the estuary, or in the lower river. We know where they're heading because we do DNA sampling on all of them, so that we can then ascribe the stock assignment to them. But in all cases, you can't identify sex physically at that point. And even in the lab, it takes several weeks before, or at least 2 to 3 weeks before you start seeing those sex characteristics emerge. So we wouldn't have to take blood on them in the lab, because we could destructively sample them when the experiment's done and look at their gonads. But in the field, you have to have that blood sample.
Hi, Scott. It's Corey. I have a question for you.
Hey, yeah.
Hi. You're doing genetic work, which is great to hear. I'm curious, are you looking at any highly expressed genes that may be associated with any of those 4 hypotheses you've kind of highlighted as maybe a culprit that maybe help find out if there's one more contributing than another, per se? Right.
So we do genomic work in some of our studies. Yeah. Many of these studies that I'm talking about here, the genomic works that we looked at, we are trying to identify which particular genes might be correlated with their survival. Generally, these have to do with immunosuppressive genes, to do with cell damage genes. Right. Some cases, they're osmoregulatory genes. So it's a whole suite of genes, and they seem to vary. But when I say we did genetics on them, that mostly did look at stock ID. But we do genomic work on some of those studies, but not all of them, just a few of them.
Yeah, no, it's complicated, of course. It's a very crazy dynamic in the genetics. I know with working with Dr. Heath in the past, looking at rainbow steelhead symmetry, and are they different? Are they the same? And we looked at very highly expressed different genes, and we couldn't find very, very much on that too. And I know it's a challenge and needle in a haystack a lot, but just figured I would ask the question.
Some of them, it's a good point. And we have tried to identify, and to some degree, we've identified, depending on the local conditions, if the temperatures are really warm at the time of capture, you might expect to see heat shock proteins that code for that as being important. But in some case, in many cases, what we're trying to do is capture fish under as benign a condition as possible before we tag them and release them into hospitable environments or into hospitable lab experiments. So when you take your samples at the beginning, these fish are quite healthy and not necessarily expressing anything that would suggest that they are impaired.
Right.
We do sample them towards the end of their life in lab studies, and that's when you really start to see these genomic differences emerging in terms of them being, highly stressed, immunosuppressed, and so on. But we can't tell that at the beginning.
That's really interesting stuff. Yeah, that makes a lot of sense. Yeah, that's great. That's very interesting work.
Oh, thank you.
Yeah. Another question that came through, was that Jennifer? Was that from you? I can't recall. I can't see there's a chat question there.
So I say, is the mortality ratio of females to males during migration occur only in salmon? To my knowledge, nobody's looked at this in any other fish. We've reviewed the literature quite a bit to look for sort of this broader trend, and people don't really seem to look at it. And the reason, I believe, is because for most fish, you can't identify sex physically until much later in their life. And in some cases, it's even hard then. So I believe that's why it hasn't really been looked at elsewhere. Interestingly, DFO now has genetic sex ID as part of their stock ID program. So it is possible now to actually get sex ID without having to take blood. They can do it genetically, which certainly speeds things up and means there's less handling for fish if you don't have to do the blood work. But generally, most people up to that point, up to a few years ago, would have to take blood, and that just wasn't done routinely. So maybe it does occur in other species. We just haven't been able to look for it.
Size of the females getting smaller, or are they about the same as they were, say, 30 years ago?
Yeah, that's a good question. Well, certainly there's some recent publications out of Alaska that shows that the size of all the 5 main species are in decline. So it was a really good study that came out just past year. And in some species like Chinook, age at maturity is also declining. In terms of our sockeye that we were studying, we have not seen a difference in size across the years we've looked at, but I wouldn't be surprised if that's happening, or it will happen if it hasn't already, because all the conditions are right for that in the ocean in terms of poorer feeding areas and higher competition.